Boa tarde a todos. Agradeço uma vez mais estar reunidos aqui em um CEBIMário mais essa quinta-feira, depois do almoço, que todo mundo está processando. E eu quero então agradecer ao Dr. Sergio Navarrete por ter aceito participar hoje desse CEBIMário. Sergio, muchísimas gracias por aceptar la invitación. Es un placer tenerte aquí y poder descubrir un poco más sobre tu trabajo y de las cosas que vienes realizando durante este último tiempo. Então, pessoal, a apresentação do Sergio vai ser em inglês, mas todas as suas dúvidas podem ficar escritas no chat em português, não tem problema, a gente vai tentar fazer a tradução das suas dúvidas em espanhol para facilitar o processo de forma geral. Então, Sergio, te voy a pasar a palavra. Muchísimas gracias una vez más. Hoy Guka nos va a acompañar, que es el director de CEBIMário, acaba de llegar. Guka, muchas gracias por participar.
Gusto.
En fin.
Bueno, hola, pues, Juan Pablo y Guka.
¿Qué tal, Sergio? ¿Cómo estás? ¿Cómo te va?
Muy bien, muy bien. Gusto verte, te ves igual, ¿eh? No hemos,
sí, igualito,
no hemos cambiado
igualito.
nada.
Sí, no, nada, no hemos cambiado nada, ni barba blanca, ni nada, todo el mundo con mucho pelo, bueno, tú sí, yo no.
Seguimos igual, sí. Oye,
seguimos igual.
¿Quieres
un placer en verte, un placer verte. ¿Quieres que dé un rápido overview de Sergio? Porque Sergio es muy mayor, estuvo aquí hace milenios y la gente ya no se recuerda de él, pero puedo hacer una pequeña presentación. Yo no sé, esto va en inglés o en español, va un poco igual, yo creo que no pasa nada, ¿no? ¿Puedes seguir, Juan, para introducir un poco?
Claro, Guka, voy a entonces dejarte que tú lideres este seminario hoy y bueno, preséntalo y no hay problema, ¿vale? Y después me avisas y le pasamos la presentación a Sergio.
Muy bien. ¿Vale? Bueno, pues es un placer tener aquí a Sergio, ahora de manera remota. Hace mucho tiempo que no nos vemos presencialmente. Esperemos que eso ocurra en breve, o por lo menos no en el año 2030, un poquito antes quizás. Bueno, Sergio es empezó en el mainstream de Rocky Shore Ecology de la costa al este de Estados Unidos. Él fue directamente supervisionado por Bruce Menge y ahí empezó su carrera profesional, creo yo, ¿no? Entonces trabajó con varios aspectos de varios procesos que regulan comunidades benthic en el intermareal rocoso y, siguió después para un postdoc, creo yo, con Steve Gaines, luego más con ecología larval y cuestiones de dispersión, conectividad. Y, bueno, poco tiempo después ingresó en el ECIM en la PUC Chile como director y hasta ahora estás como director del ECIM, creo, ¿no, Sergio? Muy bien, entonces, así es. Entonces, es un placer, Sergio. Trabajó en Brasil con varios de nosotros y incluso conmigo, y fue un placer enorme. Espero que sigamos trabajando y colaborando en un futuro no muy lejano. Y bueno, Sergio, te dejo para que empieces tu charla y luego ya tendremos tiempo para... Estoy muy curioso para saber qué cosas nuevas tienes para entretenernos aquí, y luego entonces haremos una sesión de preguntas y bueno, cuando quieras. Es un placer enorme que hayas venido. Muchísimas gracias, Sergio.
No, gracias a ustedes, Guka. Para mí siempre un placer. CEBIMário es como mi segunda casa. Estoy más que preparado y listo para otro sabático, así que ya te contaré. Y muy bienvenidos todos, gracias a los que estén escuchando. Como les decía, Guka me invitó a dar esta presentación y me decía qué cosas nuevas estoy haciendo. Y no estoy haciendo mucho, pero decidí contarles un poquitito. Algo había contado en un seminario en Arrayal do Cabo de lo que estamos haciendo en un ámbito particular de mi investigación y que tiene que ver con, en general, yo diría cómo abordar sistemas complejos, sistemas ecológicos complejos, como los sistemas en los cuales estamos trabajando todos nosotros, o la mayoría de los que están acá, sean sistemas marinos, terrestres, y particularmente cuando incluimos al ser humano como parte de estos sistemas, que agrega capas de complejidad aún mayores. Yo entiendo, Juan Pablo, que puedo proyectar directamente mi pantalla y que ustedes están viéndola así, ¿verdad?
Sí, exacto, ahorita está siendo transmitido, entonces no hay problema. Está todo
Perfecto.
correcto.
Voy a seguir. Si hay problema o no se ve que avance en diapositiva, me avisa y me interrumpen nomás, ¿ok?
Perfecto, gracias, Sergio.
All right. Bueno, les cuento un poquitito entonces lo que tenía preparado para hoy día, que más que una presentación en una temática en particular, va a ser una presentación general en lo que estamos haciendo. Y me voy a cambiar ahora a inglés luego de esta presentación. So I'll try to give you a kind of a brief summary of what we've been doing in this particular area of research that has become quite fascinating for me in the past, I would say now probably 10 years or a little bit more. But and I'll tell you at the beginning why I got interested in these aspects of ecology, which is not, like Guka was saying in the introduction, is not exactly what I was trained on at the beginning during my PhD. Like Guka said, I'm a traditional marine ecologist in the sense, rocky shore ecologist in the sense that I was trained as an experimental ecologist coming directly from the school of that believes strongly on the use of experimentation, particularly field experiments, to test hypotheses and theories of ecology. Back in the time when life was a lot simpler for many of us, and that we thought that by performing experiments we could solve most issues of humanity. And I'm not saying that in a way to put down ecological experiments. I feel they play a major role in advancing science in general and ecology in particular. And for those of you that might come from other areas of research, maybe not ecology or maybe not marine ecology, I want to remind you or tell you that experimental ecology is probably one of the main contributions of rocky shore biologists to the entire field of ecology, particularly with these two guys. Unfortunately, they both left us just a few years ago, Bob Payne and Joe Connell, who pushed forward the possibility or gave us the possibility to actually test hypotheses using an experimental design. And in many ways, they should be credited with putting science at the table of the big sciences and the big important sciences in the world. Experimental ecology, of course, has been since very, very key in testing, expanding, rejecting, and generating new hypotheses and concepts. And here I listed just some of them. So by all means, I think marine ecology, marine experimental ecology, and experiments in general are one of the most powerful approaches to advanced and test theories in ecology. And I want to add also that in many ways is a very wonderful way to keep us honest about what we really know about ecosystems. And anybody that has performed an ecological experiments, I think, know what I mean by that. When you make a bunch of predictions and you conduct your experiment and you realize that none of them comes through, and you get always some unexpected results, which tell us that we have not really fully comprehended ecological systems. But there is a fact that remains with experimental ecology, and I copied this from the classic paper by Bob Payne, the Keystone paper published in American Naturalist in 1966, in which he demonstrated the keystone effect of Physaster ocraceus in controlling diversity of species. And I would just want to highlight that aspect. It says that after removing the keystone species, the system went from a 15-species system to an 8-species system. That was the learning from that experimental manipulation, which inspired and lots of studies and theories, and is still one of the most useful concepts in ecology, the fact that a given species can have tremendous impact in dynamics and diversity of a species. But like I said, the fact remains that that's only 15 species, and that in the Pacific Northwest intertidal rocky shore, there are probably 600 species that coexist in any given platform. So when you think about in detail about what the experiment shows, it shows but a small fraction of the species that composed those communities at any given time. Recently, my good friend John Whitman reviewed the literature on experimental manipulations, and I think they reviewed 311 experiments published since 1961 in marine habitats, including rocky shores and subtidal habitats, and they came up with wonderful results. One of them is how many species, which area and the duration, and how many species are usually included in these experiments. And they show that the great majority of experimental manipulation includes less than 10 species as influencing a given response variable or a given target species that is the subject of manipulation. In some cases, the investigators monitor more than 100 species, but the subject of manipulation and control is rarely more than 10 species. So there is clearly a huge gap between what we are doing experimentally, what we can control through experimental manipulations, and the diversity of most natural ecosystems. Which are the limitations of including only a small subset of species? Well, besides the fact that we will know only really about that set of species that happen to occur or be manipulated within our experiments, and we can say very little about all the other ones that are also in the same ecosystem, there is the problem of propagation of these effects through the web, way beyond the species that conform our experimental system. Peter Jotzis published a paper in the 90s, in the early 90s, about the indeterminacy of a species interaction, and again in 2002, showing very neatly that if you manipulate a given species, the effect that those manipulations and those changes in abundance of a given component propagates through the entire web of interactions in a manner that makes it almost impossible to predict not only the magnitude, but even the direction of change of other species that conform that system. Should we care about biodiversity? Should we only care about the most important species? I think that I don't need to emphasize that, yes, we do. We do need to care about biodiversity for many reasons, but also for our own selves. And I just want to remind you this paper by Peter Vitusek, Hal Mooney, and Lubchenko, and Melillo back in '97, where they define which were the most important drivers of global change in the planet. And they identified the loss of biodiversity as one of the most important components of global change in the planet. And in a more recent paper by Dave Hooper, he compared the impact that we are having in the planet on specific ecosystem functions, mostly terrestrial ecosystems, the impact we are having by the loss of biodiversity, and compare it to many of the other drivers of climate change, and concluded that biodiversity loss is probably by far in most ecosystems the most important component driving global change in our system and loss of ecosystem function. So there are valid theoretical, but also very important practical reasons why we gotta care about understanding biodiversity and how our actions in these ecosystems and can propagate and eventually affect our own well-being. How can we deal with this rich, important, but bothersome complexity of natural ecosystems? The main answer from ecological sciences has been historically what we call food webs. Food webs, unlike experimental manipulations, are representations of ideally all the species that co-occur in a given area at any given time. And they basically represent who eats who, which species feed on which other species, and in that manner, the food web representations are only consumption, or what we're going to be calling here trophic type of interactions, and they represent competitions through shared resources. So those are the two main processes represented in a food web. And the advancement of network theory coming from the physics has allowed very interesting and rapid advancements in what we understand about ecological systems when we include ideally all the components of that community. So they are a wonderful tool to embrace that complexity. But back then, when we got together with several of my friends, particularly my good friend Eric Berlo, we were talking about whether this was a correct or even close representation to the real complexity of ecological systems. And since we all have worked on the effects of a species through habitat modifications, we know the concept of a species engineers, we know facilitation in marine ecosystems, we know that is rampant and critical in all ecological succession processes. We were clear that this was far, far from being a representation of true complex ecological systems. So we know that species do more than just feeding on each other, and that sometimes even when you feed on a given species, that same species can be your habitat. And this is just an example of a predatory crab that feeds mostly on mussels, but it also lives within the mussel bed. And this is common in all ecosystems where you get pollination, you get associational defenses. The trees may provide fruits for some birds, but the birds also nest within the trunk of many of these trees. Kelps provide habitat for multiple species. In fact, sometimes are treated even as a separate full ecosystem within coastal environments, and the same occurs in all ecosystems, freshwater, marine, and terrestrial, where species interact in manners that cannot be represented by a food web of who is eating who. So there is clearly, or there was clearly, a need for integration. I believe that there is still a lot to be done conceptually to integrate the trophic type of interactions and what we're going to be calling simply as non-trophic, all those interactions that are not getting part of the biomass of someone else or another species. So Eric Berlo put together a really nice team of theoretical and experimental ecologists back in 2009 at Yosemite to just think about how we can put together a sister eating mussels from the kelp, and the kelp providing shade for chitons or other species. And I think that one of the interesting outcomes of that effort was a conceptual approach to trophic and non-trophic interactions. And it was quite simple. It was the realization that species are in the representation of a network are nodes that need to capture biomass, which we call consumption of a given biomass, if you are not, of course, a plant or any autotroph. And once you consume that plant, you transform it and assimilate it and produced some of that biomass into new offsprings, and some of that biomass allows you to capture more resources and live. And that the non-trophic interactions in a network will therefore affect one of two components, will affect the rate of consumption of a given species, or will directly affect the way in which the energy that is consumed is actually processed within the node. So we separated between non-trophic interaction that affect feeding, which has been the focus of quite a bit of theoretical research, and those non-trophic interactions that affect non-feeding attributes like assimilation rate. A species under a shade in a desert assimilates at very similar rates than a species that is out there in the sun under heat stress. That was I think, one of the important breakthroughs of that workshop. And also we proposed a very simple way to model these interactions in a real network. And the approach comes simply from an extension of Jotis and Inest resource biomass consumption models, in which each component of the energy intake of a given system, this is written here N, but it's actually the biomass transformation function per unit of time. So the income of a given species through consumption could be modified. All these functions that establish the dynamics of the biomass of any given node can be modified by a third species. And I'll tell you a little bit later what we've done, particularly Sonia Kefi has done to actually apply the model to a real web. So it's not really anything too new in terms of the approach, except probably because unlike the Jotis and Innes original model, we added an immigration and immigration process, thinking that in many cases, like marine ecosystems, non-trophic interactions are critical to get a species established in a given system. So that was what we did in terms of trying to put together conceptually trophic and non-trophic interactions back then. And then we were faced with a challenge that I had not undertaken, quite frankly, which was, well, can we construct a real ecological network, a network where we know which are all the trophic interactions and which are all the non-trophic, or at least most of the non-trophic interactions. And like I said, I would have not really been involved in that project, but Sonia Kefi and Evie Withers convinced me that it was possible, that we could work on putting them together if we knew a system in which we had a very rich history of experimental manipulations. And why we need experimental manipulations to build an ecological network is basically because the non-trophic interactions are not binary. They are not simply prey or predator. I mean, when you deal with food web trophic interactions, a predator either eats the given species or it does not eat it. In the case of non-trophic interaction, they generate that additional problem that the effect to deem that a particular interaction exists is a function of the abundance of those species that modify the other one. So basically, a tree does not make a forest. A forest has tremendous impact on the rest of the fauna and flora that inhabits that given area. But if you put the same species but only one tree, it will not have such an impact. So clearly, non-trophic interactions are non-binary and require demonstration that a given species does have those effects in the community. So we decided to go to one of the best studied systems in the world, probably, in a small area, which is the Marine Lab. I'm sitting right in front of this place here. We have new buildings now. Guka, you should come and visit again. We've grown quite a bit in the lab. And in this sector here, which is a marine reserve and surrounded areas, there have been probably 200 scientific articles published since the early 70s, late 60s, mostly by Juan Carlos Castilla, Bernabé Santelices, Patricio Ojeda, and many other professors that worked in this particular section of the coast. So we thought that this is probably one of the best studied rocky shores in the world. There are other ones too. And also the level of diversity is relatively moderate. So we would have loved to tackle the Pacific Northwest, but the number of species is huge in comparison to central Chile. So this seemed to be a little bit more doable for us. And we still have the hope to eventually construct something similar for the Pacific Northwest. And this is what we did, or I should say what Evie and Sonia mostly did. Sonia Kefi from Siena West, France, and Evie is a colleague here at Las Cruces. And this is the food web representation of the rocky shore of central Chile with mostly algae down here. And in this case, filter feeders are represented a little bit higher up in this web, and I'm going to get back to it if I have a little bit of time. And then top predators up above. This is just to remind you that in Chile, like Brazil, or probably even way more than in Brazil, the rocky shores are heavily exploited by humans. So there is an actual fisheries, artisanal fisheries that exploit at least 14 species in these systems commercially, and those are marked here with a different color. So this is who it's who. This is the food web of the rocky shore, and this is what we did through experimentation. We added the non-feeding positive links, which are not many, but they exist, and then finally the non-feeding negative and non-feeding positive links in this ecological network. So there you have it. That's the ecological network of the rocky shore of central Chile. And as far as we know, the first one that is being represented in the literature. So it's by far not complete. This is absolutely incomplete. But we thought that we had reached a level of information that allow us to at least say this is the way an actual network of interactions looks like in nature. And I am sure and I hope that this is continually revised and that many of the links here might prove to be not true or not important, and many other new ones might appear in this system. So with this, for instance, and I put there the web address where you can get in and play with this web is publicly available. You can represent or look at for hubs of interaction of trophic or non-trophic interactions. And for instance, here we illustrate if you want, a species engineering role of mussels that form mussel beds in intertidal communities. So after that, unfortunately, we did not, and I still don't have another ecological web to which we could compare to. But we still thought it would be interesting to determine if there was some structure. Is there a structure in this entangled bank that I just showed you, or this full set of interactions are relatively random in terms of the way species interact with each other now in what we call a multiplex network, which is links that are trophic and links that are non-trophic positive and non-trophic negative. And Sonia Kefi with Vincent Millet from France and several of us started to think of ways to summarize the structure of these webs. And that required a little bit of developing slightly different algorithms to assess compartment, which are based a little bit on modifications of the modularity index, very commonly used in ecological networks in general. And we first determined that there is only a subset of possible interactions that are actually realized in the web. So of the entire set of possible interactions, only a subset actually occurs in the intertidal web. That most species interact only in pairs, meaning they establish with a given other species only one type of interaction. That only a subset interacts with another, with the same other species, not only in through one type of interaction, but through two types of interaction. For instance, the prey and habitat at the same time, and even fewer species interact in all three ways, meaning having positive non-trophic interaction, having negative non-positive interactions, and also being predator and prey at some point in their lives. The other finding was that despite the complexity of the web, there were only 14 multiplex nodes. And those multiplex nodes are the ways in which species interact through these three levels. So there are compartments within the web, and the compartments are far fewer than the over 106, I think, species that conform the intertidal web. The way they interact, these are the trophic, non-trophic, negative and non-trophic positive interactions among the nodes. So the different nodes not always connect in the same intensity through trophic and non-trophic interactions, but those nodes are the groups of species that are most similar in the way they interact with each other. Not only because they share prey, which is usually the interpretation for food webs, but because they share prey and at the same time they provide habitat or they have positive effects on similar species within the rest of the community. There is a part there that we also did in terms of trying to determine whether that particular structure was more stable or not than a random distribution of the different types of interactions, and our analysis showed that probably it is. It probably is a more stable structure than what you would expect by random. What we've been doing more recently is trying to determine spatial variability in networks, and one of the issues that still is, I think, very much in infancy in network theory is really understanding how spatial component, how to incorporate a spatial and temporal variability in networks. And networks are in many ways stationary representations of a species interaction for a given system that usually represent an undefined area or community. And while more complete, the best is representation. And that creates some problems conceptually, because if you want to represent all the interactions of a given area, you want to have more and more time to examine who is eating who, and also having a larger area to represent all the species. And creates a problem on how then you analyze a spatial variability. And to me, that's one of the breakthroughs that is needed to be made in order to go from network approaches to more applied issues. But what we've been doing empirically now is trying to examine how the different types of interaction vary across the space empirically, not theoretical models. And what we did was to use our surveys. We have surveyed the coast of Chile, including not the coast of Chile, a section of the coast of Chile, but it's still about 1,500 kilometers. And across this gradient that I show you here, from Temblador over there, all the way down to Buchupureo in south, for reference, we are right there in the seam, about the center of this region. Across that gradient, of course, there is a lot of variability in environmental conditions, and one of the main drivers of environmental variation is upwelling. The upwelling of cold, nutrient-rich waters near the shore, which creates differences in nutrient loadings, in temperature regimes on the shore that can affect all these species. And we examine to what extent the trophic component, all the trophic links within the web, the positive non-trophic and the negative non-trophic varied across this regional scape, and which were the most likely environmental variables driving those changes. And very briefly, the main results show that that the least sensitive type of interactions are the trophic type of interactions. So our trophic components of the web are less sensible to environmental variability than the non-trophic interactions, and that the most sensitive ones, the ones that change the most, are the non-trophic positive interactions. And the main driver, potentially driving factors, this is only correlations between network structure and environmental conditions. And the most likely factors driving that variability are indicators of productivity, not directly upwelling, but indicators of water productivity, and indicators of environmental predictability. So more than the direct variables like mean temperature, it was indicators of the variability in those environmental variables. One of the things that we are also working on is trying to actually get to the complexity of a real network. And when I told you, I presented to you the real ecological network of the intertidal shore of Chile, is not really the real ecological network. There is a node there. There are many deficiencies, but one of them that is obvious is that one of the nodes that I show you in those figures is biofilm, just like that. And that is the way that is treated in pretty much all reported marine networks in the world. There is about 50 now that have been reported with quite detail. And in most cases, there is a little node, maybe two, that say biofilm. The problem, as you know, marine ecologists, and in Brazil, there are several ecologists, marine ecologists working on biofilms. So Ronaldo has been working for many years, also Guka, on the importance of these biofilms. Biofilms are, by themselves, a hugely diverse community that is actually way more diverse than the entire rocky shore communities of macroscopic organisms. So there are 3 different realms that compose the biofilms, including fungi, bacteria, archaea, and that we were probably underestimating the complexity of a true or realistic ecological network. So one of my students, Clara Arboleda, decided that it was time enough to start resolving a real complexity of ecological networks. As you know, the study of biofilms goes back in marine system to the 60s, but only, I'd say, the past 10 years, the development of molecular techniques at low cost has allowed us to really study the composition of these communities across sufficiently large number of samples and conditions. Before, we could not really know which were species or even major groups within this biofilm, and most of the studies focus on only the photosynthetic organisms that compose these biofilms. Before I show you some of the things that Clara is doing, which I think are fascinating, just a caveat on microbial co-occurrence networks. For those of you that are working with microbial communities, microbial biofilms, the typical approach to construct networks in microbial communities is through co-occurrence matrices. So you looked at the occurrence of ASSB or OTUs across a space, and then you construct a matrix of interactions based on the occurrence patterns of these units, taxonomic units, which of course creates several problems because they might not necessarily represent the same things that we macroscopic ecologists talk about when we say a given interaction between species. And we relatively recently did a study using our intertidal databases on co-occurrence patterns and knowledge of which species interact from the network that I just showed you. And the main conclusion is that we got to be very careful when interpreting and comparing networks constructed through co-occurrence matrices and network constructed through direct observations or experiments to determine that species actually interact and in which way. Not necessarily one is better than the other, but it's a little bit of apples and oranges when it comes to trying to compare those two things. So because of that, Clara actually had to develop a number of experimental methods, new experimental approaches to examine the interaction between macroscopic organisms and these microbial organisms. And that has been really wonderful to see her develop a number of techniques combining manipulations and molecular techniques and microscopic techniques that were not available when I was a student and not available because of costs just 10 years ago. And I think that is being, or it will be a major breakthrough in our understanding of macro and microscopic world. This is very preliminary still. She's been focusing on 5 different species of grazers that belong to different major taxonomic groups. And these are just some of the main, I think that these are by orders of microbial groups. And you can see now that the different grazer species affect these different microbial groups in very different ways. Some of them positive, like she's evaluating both the trophic and non-trophic positive and negative effects that these organisms have. So it's probably going to be, I think, one of the first representation of actual network of interactions between, this is only 5, but at least those 5 grazers and the microbial taxa, microbial units. We haven't found yet a good way to represent the effects at the level of the OTUs because there is about 1,000. So we need to find good ways to represent the information. That, like I said, is still an area that I believe needs a lot of work, and Brazilian shores are ideal for that, actually, because the biofilms there are quite really huge biomass in comparison to Chile. And there is a lot to be done there. It requires combining expertise from different areas. Just to end, a couple of things in terms of theoretical approaches. To me, like I said, one area that needs major breakthroughs is how we develop tools, conceptual and theoretical, to study spatial variability in networks and temporal variability. So together with Pablo Marquet and Rolando Rebolledo, who is a mathematician of open systems, quantum mechanics, we developed like a stochastic open system version of the model that we had developed with Sonia and colleagues back then, which basically now deals with nodes that are open to exchange of biomass from the same and different systems. So we use the analogy of a meta community and model what we call the local community as an open system that is constantly exchanging biomass through 2 types of processes, Brownian processes and Poissonian type of processes, thinking that you get variability in environment and interactions that occur at a given time scale, but also variability because this system that you're modeling is embedded in a much larger system that is exchanging biomass with your local community, and that may occur at a different time scale. So to me, it's a very promissory approach, and we are working on trying to make these more digestible ecologists, including myself, so that we can actually apply to address particular ecological problems. And the other area, and this is the end of what I'm wanting to show you, is we are starting to try to make the connection between this complexity of ecological systems and artisanal fisheries. In Chile, you probably know, Chile is one of the main exporters of shellfish and algae in the world. And in South America, we even surpassed Peru as exporter of coastal fisheries, not the industrial pelagic fishery, but of coastal fisheries. Chile is the number one exporter. And I say because many times when you mention artisanal fisheries, people think of very small scale subsistence type of operation. These are commercial, quite big operations that can have major impact in coastal ecosystems. So it's not that concept of only local consumption. I believe 90% of the species that are exploited or more are actually exported. They are not for local consumption by what we call artisanal fisheries. So one characteristic, of that fishery is that they exploit multiple species. And in the intertidal zone alone, they collect at least 14 that are commercially exploited, and we estimated that at least 10 more are locally consumed, and they are not part of any records. So we've been thinking, how do we know what is the impact of this exploitation in local communities? And we are using now, this was pasted wrong, so it's not that complicated of a math. It's simply the same model of biomass transfer that is applied in what they call allometric trophic network approaches. So trying to parameterize a given network of food web, only consumption in this case, with what we know about basal metabolic rates and requirement by organisms in general. And since we have body sizes and we have estimates of biomass, we can parameterize these models with realistic patterns of energy requirements. And that now allows us to get a glimpse at propagations of effects in a given community. And this is a paper that was produced by one of my students, Isidora Avila, and my colleague, Fernanda Valdovinos, who is now a professor at UC Davis. They've done all this work, really. And one of them, the first report that she just published is showing the relative impact of variation in productivity versus fisheries. And I'm not going to extend in this, but what this figure is trying to do is summarize the impacts that we observe in those food webs. And don't get mistaken, these are top predators, carnivores, omnivores, basal species, fisheries, but the modeling behind is not modeling these groups, it's actually modeling the species that are affected, that then we integrated their biomass to examine the effects at the level of those groups. So it's not the typical path analysis type of approach, but it's actually modeling the food web. And the conclusion is, of course, that fisheries is having a very large impact on non-fish species, at all trophic levels. And interestingly, that it is the food web is more sensitive to changes in productivity than actually the fisheries, despite all the fishing pressure. What we're doing now is actually bringing some realism to the way we address the fishing impact in these communities. So that's I know I extended a little bit more, Juan Pablo, than you probably expected, but I'm going to end it there. I hope that we get some questions at least. Sorry about the extension.
Bueno, Sergio, muchísimas gracias de verdad por tu charla, genial. Voy a colocar a Augusto también aqui para participar da discussão. E, bueno, pessoal, vou abrir então a sessão de perguntas. Agora, quem quisesse deixar suas perguntas, pode deixar suas perguntas aqui no chat. A gente está, o Augusto e eu podemos ler as perguntas para o Sérgio. Eu vou parar de compartilhar a sua apresentação, Sérgio, para abrir o espaço para perguntas. Então, enquanto o pessoal...
Sim, para falar, Juan. Eu não tenho aqui acesso ao chat, mas eu posso começar, talvez, e o pessoal depois entra. Sorry, I'm talking in Portuguese. I'm sorry, Sergio. I know you're a little bit rusty with your Portuguese, so maybe we can go back to English again. And I can start with a question session. Well, first of all, amazing work, Sergio. Yeah, lots. I knew some of those papers already. I had a first reading of a few of them that you presented, and but it's overwhelming, the work that you're doing there, and I suspect that you will end about doing something like that, Sergio, because you are a fan of complexity, and this is nice because, of course, systems are really quite complex. But I was about there are many different questions, but this will take us a full day, so this is not the case now. Maybe we can catch up later and have this conversation on some curiosities, Rocky Shore specific things which are so funny and so forth. But now I would like to perhaps go to a more general question, and I will take perhaps the lead of your collaborator Eric Berlow a while ago, when he was one of the first guys trying to tackle up this complexity, mostly on Rocky Shore systems. And it is true. I totally agree with you that maybe that we were addressing main changes by manipulating a few set of species. But trying to look things in the other way around is that, all right, this is true, but there are a few species that still they can manipulate what we call foundational species, which in turn they will translate through facilitation processes to this huge diversity that you mentioned. So it is true we have like a few hundreds of species in the rocky shores of Northeastern America, but it is true that if you take the foundational species, this will drop by a half or a third or something, because so yes, of course, still this is just a plea to say, all right, so there are still a few species that are worth looking at, right? And the second thing is that yeah, it is amazing how things can change if you look at a more complex way. But still, when you put all this chart of these energy flows and links and everything, it is true that if you manipulate a few of them, even the ones that are not obviously important, you can have some very complex responses. But if you go to the end of the line and to the actual community, so communities naturally they change on, let's say, or they can vary in a bunch of a few options, right? So you have like discrete states of many different communities, and in spite of all this diversity and all the possible interactions, this will end up on 3 or 4 or 5 different scenarios, right? And this is really interesting and reminds me of that talk of Eric Berlow about that, that, all right, so complexity actually can lead to simplicity, because they will end up somehow, these processes will cancel out or whatever it takes to make like this, that the end of the line will be like 3 or 4 or 5 or 6 different states in a community, despite of all this complexity. So what do you think about this?
Well, I mean, the thing that is true is that there are patterns in the complexity, no doubt about that. And we didn't need all this modeling to show that there were patterns of complexity, because we know that despite the fact that our predictability, our capacities to predict changes are very limited in general, still things are recurrent. Still patterns exist in nature, even when you deal with full sets of species, not only the dominant ones that are obvious to anybody. So patterns exist, and the three questions, I mean, I think that the critical question is, can we understand enough of what is driving patterns in complex systems that we can reproduce them conceptually, theoretically, and ideally anticipate a given change? To me, I'm always been a person that is more question-oriented than a modeler, in the sense that by creating these representations, I see several colleagues trying to go towards representing the actual real network. And that is not my goal, because it's still a model. This is not real. This is a model that is as useful as what we can answer, which type of questions can we answer. So to me, using the models is a way to get a realistic representation of complexity, so that using these tools, we can anticipate some of the changes that we cannot study really by focusing on any given species. So to your, I think, reflection on there are some species that are important, there are some manipulations that lead to major changes. I think that is true. I mean, and that's why this is not really a better approach to science or to ecology by no stretch of imagination. I think it's an approach that allows you to answer some questions that you cannot answer by saying, if I remove the sea star, I'm going to lose 7 species in the system. And then, because that is very important, and it's a keystone effect and allows coexistence over a shared limited resource. But the question there remains, what happens with the other 455? Have they been affected or not? Are they going to be randomly getting in and out of the system? Can I assume that they are completely random? Most likely not. Most likely that effect will have some impact on at least a subset of those 450 species. That is a question that in no way puts down the findings of the experiment. It only puts the limits on what kind of things that I can answer by that experimental manipulation. So in this, as much as in what I do, I work a lot on dispersal and in combination of, as you know, numerical models and particles and a little bit of empirical data. There are still representations, and we want them only to be realistic, not real, realistic of complexity. So my effort to get into the microbial world is because it's a component that is so diverse and I think so important that it moves at time scale so different than the rest of the macroscopic organisms that we need to use this type of representation to at least anticipate patterns. We are not going to be able to do it because you do these manipulations with microbial communities that are tiny. So it becomes a real issue to say, well, what would happen if I simply remove all the biofilms of the entire shore? That cannot be done. And as you know, the issues of scale when you do manipulations are inherently associated to complexity and also diversity. Long answer to say yes, Guka.
Yeah. Okay, yeah. Fantastic, very good.
Okay, Sergio, you have the several questions here in your chat. Maybe Guka can read in this question. I can try reading this question. I have one question about your presentation. Basically, I wondering how do you include, for example, the different traits or try to explain the different network you have? Because I understand, for example, I expected different traits influence different force when you have the trophic networks. And how do you think this is possible to include traits for these different species?
Juan Pablo, are you talking about traits like morphological traits of the species, or?
Yeah, you can use it morphologic, you can use it behavioral traits or ecophysiologic traits, for example. I don't know, it's possible how the tolerance you have one species when you remove or you include more temperature and how the network outward for these chains.
Yeah, there is an inherent limitation in the approach, like I said, that is associated, I think, to the problem of using it directly with spatial variability or temporal variability. Because you don't need to, but the way we've been doing it so far is usually representing a node as a homogeneous taxa. And in many cases, you could, and I'm sure it's been done, you could say, okay, the species takes 2 different forms, and you could add it. You could say, okay, nodes are not all the same; they represent different stages in the taxa, and one with a given attribute, the other one has another attribute, and so on. The problem is that in general, the nodes don't evolve in networks. So even if you include that complexity, it would be one step forward only of complexity, saying I'm going to now model beginning stages, recruits, and larger organisms. Or the response of a given node is not going to be uniform. It's going to be a node A, which is a species in condition A, species in condition B. If those are relatively permanent over the dynamics of the system you're modeling. But still, there is not, in my mind, not a satisfactory way to include the evolution of the nodes in those networks. And I say that knowing that Miguel Lurgi, one of my collaborators, has like 4 papers on that, on evolution, on network evolution with, I believe, Leroux and Montoya. But I still find that the approach is quite like a cartoon of how nodes evolve and respond. So I think that that's another problem. How do we get the node that by its own evolution is rewiring the entire network? And that is what happened in many systems. You change the quality of that node, and it's not only changing assimilation and transformation of biomass, it's changing the way it is wired to the rest of the components in the network. There are people working, I think it's from physical mechanics, I'm not sure which area, and it's a hot topic, rewiring. In fact, we got the comment recently on a paper by one of my students that the author wanted to see rewiring. It was a software we were writing, and the person wanted us to include rewiring conditions. Well, like in ecology, it be like we are so far from knowing which are going to be the processes that now lead to reconnecting to different having established different types of interactions when my own conditions change, and that to me is still far ahead from my point of view.
Thanks.
Yeah, just adding a bit on this, because it's this kind of new papers coming up showing that, all right, you have some species losses and people are trying to predict what's happening in communities after those species losses, but it has been shown for a few guys that rewiring is possible. So sometimes a function was there and it was not lost when the species went off the system, because others will play the same role and they rewire the system in a very different way. So I read it once, and the first time I was a little bit skeptical, but then other papers followed with very, very similar conclusions. And yeah, it seems that rewiring is way more common than we could suppose at the very beginning. So yeah, very interesting. Rewiring.
Well, Sergio, Guka, you want reading the question, or you prefer I reading?
Well, I don't have the access to the chat myself. I cannot find it here in my screen. If you want
the part of the comment, you can see all the question here, but I don't worry, I can read in the question. You have several questions here. I start with Karina Emmer. Yeah, he's wondering, is it possible distinguishing biofuel of the positive or negative interactions? And we're expecting different microorganisms will perform different interaction effects.
Yeah, with Clara, like I said, I did that parenthesis because usually people working with microbial communities or microbial networks, they build them experimentally or through surveys, through sampling over space. And by sampling over space, you now build the given interaction based on co-occurrence. So the species that are there using, probabilistic models or simple correlation matrices or something like that, or maximum entropy also, things that allow you to try to get a glimpse at probabilities that species are together or not, basically. And then you build that network. Well, that way to build a network definitely does not allow you to put directionality of the effects. So you do not know negative or positive. It becomes because you don't know which direction is occurring. You only get that they are negatively associated and things like that. So there is a bunch of restrictions. That is why Clara developed an experimental technique to test that in the lab, which is basically traditional experimental ecology taken at the level of the microbe. And she had a battalion of, I think it was 16 undergraduate students working full time for 2 or 3 days, running a huge experiment with all conditions replicated with the 5 species. And unfortunately, I couldn't find the photos before the seminar to give you a glimpse of what it took, that experiment. But the list of acknowledgements is like 16, 20 people there that all participated in performing the experiment. The experiment, fortunately, was like only a day or 2 days long. It was a grazing experiment, but conducted like that because we needed enough replication to do it. So basically, in that case of what she's doing, she's getting back to traditional ecology where you get a control and you get a grazer, which is the impacted, and she's using indices that Guka knows well, and some of you of interaction strength. I worked on that in my graduate studies, and she's examining both the trophic component when she allowed grazers to graze and the non-trophic effect, because mollusks have a microbiome, and the microbiome is external to the food, to the body. So what happened is very different to what the type of interaction that we know in microscopic organisms. These mollusks are moving around and they are exchanging their own community with the free living biofilms. And even though there is a few groups that are exclusive and specialist of a given species of mollusks, many of them are not. So you put them on a community, and it's what they call community coalescence, because it's basically like the Isthmus of Panama. You get thousands of species that are being transported everywhere, that are in constant interaction with the biofilm that is being also eaten by the animal. And they have an impact. And we call that the non-trophic interaction between these grazers and the free-living biofilm. So she quantified those things, and that's why she was able to detect the trophic positive, trophic, and the non-trophic positive and negative. So it's not a novelty in terms of what you do to quantify interaction. Is what we've been done for 60 years, is that you need to develop a technique that has several limitations, but is reproducible, so that eventually someone can repeat the things in a similar fashion, so that to see whether results are similar, different, and so on. So it's becoming developing approaches that other researchers can do, basically in a similar fashion. And for those, just the last thing, sorry, for those working with microbes, they know that just the surface is a major issue. Things that we don't even think about with microscopic organisms. The microbial communities interact with the surface, with the chemical properties of the environment at a scale and time scale that is unthinkable of for microscopic organisms. So you need to develop a technique that is reproducible that way. Sorry for the long answer. Another one, Paulo. I hope it helped.
Yeah, don't worry. Okay, I continue here reading the question, and we'll talk Ryan question with you. You note the intertile is high dynamic environment between in the tiles, the fat, mostly the intertile species are inserted in two words. In most cases, field words are available to investigate and collect for logistic reasons. And your expertise, what kind of the change you releasing to a current biological interaction with the tea flow, the intertile rocky shores?
I didn't quite get. Could you repeat the last time? I didn't understand.
Yeah, so you can read the question. Is the part in the comments, and you can read it. Ryan, the question is,
Ah,
in your expertise, what kit of the chain you release to occur in biological interaction with T-float, the intertal rock shores?
Yes, I find it here. It's 225. It's 225. The comment is: in your experience, what kind of changes do you realize to occur in biological interactions when tide floods the intertidal rocky shore?
Yeah, I'm not sure whether if you could or he could. I'm not sure whether it is the idea when you get tides up or down, how the interaction change at that time scale, or whether the trophic or the non-trophic interactions change with the flow of the tide. I'm not sure I understood. What we represent are relatively static in the sense food webs are pretty much static, representation of the interactions. In the case of non-trophic interactions, they're still static in that the only thing that varies or that makes a difference in terms of whether the interaction occurs or not is the biomass. So once you deem that there is a potential effect of a habitat, you say that doesn't change with environment. What the environment is doing is changing the biomass, and with that, changing the importance or existence even of that non-trophic interaction. We are not at this point working on interactions that are changing the nature of the interaction environmentally. So if the environmental condition is directly altering a positive to a negative type of interaction, we are not dealing with that, which is, you can think of systems in which the nature of the interaction is changing depending on the environment, I suppose. No, I'm not entirely sure that is close to what was asked. Sorry about that.
Yeah, Ryan has another comment here. Maybe Guka reading, or I don't know. Anyway.
Yes, well, I have. This is the one. Okay. Well, I see one here about carrying Emmer at 225 as well, and also about the nodes as biomass exchanger. How can one estimate that for positive mutualistic interactions? Did you get the chance to think about that already? So I think it's about biomass exchanges at nodes in positive mutualistic interactions, Sergio. Not an easy one.
In what we model, once to model the dynamic of that system, remember that the non-trophic interactions are by definition not direct exchanges of biomass. So what we conceptualized before was that all the non-trophic interactions do not involve direct transference of the biomass in the system. That all the transference of biomass occurs through what we call the food web, the trophic type of interaction. And therefore, all the positive or negative are altering some component of the acquisition or altering the way I process that biomass, nothing else. So that was a little bit of a breakthrough. It's very simple and obvious, no? But it's a little bit of a breakthrough to then say, okay, I can address these issues because I could think of functions in which having a forest decreases the capture rate of a lion on the springbok, because the springbok now seek refuge in the forest. And that is going to make that holding, which is what we've done, that disk equation of functional response, changing the parameter that has to do with the search or the capture rate. So now that searching or capture time of the functional response of the predator is a function of the biomass of trees. So if there are very few trees, you get, it's not that the capture gets infinite or that capture rate gets zero if it was the other way around, it simply reaches the level that we usually define in a food web, okay? But if you have the forest, then the capture rate is going to decrease not to zero, but it's going to decrease to nearly zero in some cases, thanks to the fact that you get refuge provided by a third species. That is the way we've been doing it. So there is no direct exchange of biomass from mutualistic or facilitative or interference competition. So interference competition, which is so common in marine ecosystems, is not represented in food webs. Is represented in these new approaches where you get given a space and direct interference between species using that resource. I'm not sure whether that helps either. Sorry, kind of complex questions, Guka.
Well, you have in Spanish question. I changed again the idiom language in this moment. Gracias por los increíbles conocimientos en teorías ecológicas, Casilda Rocha. En un paisaje recifal de menos de 1000 metros cuadrados, ¿cómo podemos generar biodiversidad de grupos tróficos de peces arrecifales?
Cuando dice cómo podemos generar, no estoy muy claro si... and I'm going to change, I guess, to English because it might not... Sorry.
No worry.
I'm not sure how we can generate diversity of trophic levels in the reef. I'm not sure whether that question is whether we can manipulate diversity. In those type of systems, I'm not sure this network approach would be what I would follow. I personally would say, if I wanted to manipulate, whether there is an indication of habitat limitation. And if there is an indication of habitat limitation, manipulate habitat, which is commonly done. But not in all systems, habitat is a limiting factor. I'm not sure I would use this framework of network for those questions.
Yeah, maybe I can talk more about reef fishes. Is more my expertise area in this case. And well, it's complicated. You generate new species or new trophic relationships when you seed for reef fishes in general form. I think it's impossible when you have the small area. I think this is not possible in this case. Well, thanks, Sergio, for your talk. It's very, very congratulations. It's very good talk today. And I finished this seminar because it's the time in this moment.
Yeah.
And we talk more con Guka and another place en this moment. Y bueno, muchas gracias nuevamente por la
Gracias,
oportunidad.
Juan Pablo. Gracias, Juan Pablo. Gracias.
Pessoal, muito obrigado a todos. Muito obrigado a todos que ainda estão participando. Eu sei que ficou confuso mudar de 3 idiomas ao mesmo tempo, mas peço paciência para todos. Obrigado, Guka, também por ter participado dessa oportunidade de conversar mais com o Sérgio hoje.
Obrigado, eu.
Eu quero convidar, então, a todos que estão nos ouvindo ainda para o nosso próximo seminário para comentar um pouco sobre cenário global e tensões ambientais. O que será da sociedade de amanhã? A gente vai dar uma mudança total de assunto. E é isso, pessoal. Eu espero vocês na próxima quinta-feira. E thanks, Sergio, again for your talk. Very good.
Thank you very much. We'll talk soon, Guka.
We'll catch up later, Sergio, of course, about many different things, including the Católica. Who knows?
So go have a good lunch, guys. Bye-bye.
Yeah.
Bye-bye.
Bye-bye. See you. Bye-bye.